Hello everyone, we are now going to go over the peripheral blood flow cytometry plots from a 50-year-old man who presented with lymphadenopathy and atypical lymphocytosis, which means there are circulating atypical lymphocytes in the peripheral blood. So this is CD45 versus site scatter. We see that majority of the cells are in the lymphocyte gate that is defined by bright CD45 and low site scatter. We have scant cells in the monocyte and the granulocyte gate. Monocytes have bright CD45 and slightly higher site scatter than the lymphocytes. Granulocytes have dimmer CD45 than monocytes and lymphocytes, and they have very high site scatter. So now let's see what these lymphocytes are phenotyping as in the subsequent plots. Now in CD20 versus CD10, we see that these lymphocytes express bright CD20. They are negative for CD10. The small spray is considered negative, and they are negative for CD34. When we see kappa and lambda, they are all lambda restricted. We don't see any kappa positive B cells. So that shows that these are neoplastic. And again, when we see the intensity of the light chain expression, we see that the lambda expression is pretty bright. If you compare this with the neoplastic cells we saw in the previous chapter of CLL, they had dim CD20 and dim light chain expression. In contrast, here we have a bright CD20 expression and bright surface light chain expression. And these cells are again negative for CD10 and 71, they're negative for 10 and 34. Now, when we go to tube 2, we have markers like CD5, CD23, FMC7, and CD200 in this tube. So this will further characterize the lymphoma. So far, we know that it is not a CD10 positive B cell lymphoma. Now we can see whether it is CD5 positive or not, and if so, what else we can say about that. Now here in CD19 versus CD5, we see a population of lymphocytes that are blue, that are negative for 19 and positive for CD5. So those are your T cells. And there are cells that are negative for both 5 and 19. So those are also in the lymphocyte gate. So possibly those are NK cells. Now the 19 positive B cells normally are negative for CD5, but here we see that they are all positive for CD5. So we now have a CD5 positive B cell neoplasm. Then we see that it is negative for CD23, it's positive for FMC7, it is negative for CD200. So these are the markers that are going to differentiate differentiate the two CD5 positive B cell lymphomas that are CLL, SLL, and Mantle. So CLL is positive for 23 and 200, and it is usually negative for FMC7, but here we see that our B cells are negative for CD23 and 200, and they're positive for FMC7. So this is a mantle cell lymphoma with circulating mantle cells in the peripheral blood. This is just a repetition. 5 positive cells are negative for 23, and again in kappa lambda they show that they are all lambda light chain restricted. So in this one, we will say there are different types of reporting. Some people are confident that this is a CD5 positive lymphoma, doesn't have anything phenotype anything phenotypically like CLL. So they might say mantle cell lymphoma, but usually for mantle cell lymphoma, you need a cyclin D1 or SOX11 positive D or positive fish finding. So we prefer to call it CD5 positive B cell lymphoma, and then put in the comment that the phenotype does not appear to be like a CLL, SLL. It is more suggestive of mantle cell lymphoma, correlate with the lymph node morphology and cyclin D1 expression, because just from flow, I don't think you can say mantle cell lymphoma unless the patient already has a diagnosis of existing mantle cell lymphoma. In that case, you can say peripheral blood is involved by the patient's mantle cell lymphoma. But here it is better to say CD5 positive B cell lymphoma. Some people say CD5 positive lambda restricted B cells identified and give the percentage. Either is fine, but if it is CLL, people are more comfortable calling it CLL, but mantle, it is better to correlate with your lymph node morphology and then call it mantle. And it's better not to say mantle just from flow. And one other interesting thing that you can see is sometimes mantle cell lymphoma it is also called the great mimicker, like, the melanoma of the search path. So they can look like anything. And also, if you remember, there is a variant called blastoid variant in mantle cell lymphoma, and those can actually be sitting in the blast gate, or they can have high site scatter because those are larger cells. But the phenotype would still be like the mantle cell lymphoma. You just have to remember that, sometimes with that morphology, you can have CD5 positive B-cell lymphoma. So in summary, mantle cell lymphomas express bright CD20. They have bright surface light chain expression. They are negative for 23. They are negative for 200. And they're positive for FMC7. And when you report them, it is better to just say CD5 positive B-cell lymphoma and give your a suggestion in the comments rather than top lining it as mantle cell lymphoma, because that's a lymph node diagnosis. Thank you.